Bismillahirrahmanirrahim. Today we will discuss about the polymerase chain reaction or PCR. So basically PCR are a process in which we can make multiple copies of our desired DNA sequence. Now let's go to start to explain the requirements of PCR, that what requirements are involved to do PCR process. So the first instrument is a thermocycler, which we can add our sample or a template of DNA. Now the second uh, basic tools or requirement for PCR process is a template strand. Though template are basically those strand which we want to amplify. The third number of requirement is basically a primer. So we know that primer is basically a short sequence of nucleotides which are responsible to attach our DNA sequence to help in the amplification process. Now the uh, fourth number requirement for the PCR is take polymerase, which are basically derived from take Thermus aquaticus bacteria, and cofactor are also used for this process, like magnesium ion. They provide stability, and water buffer solution and DNTPs are required for the PCR process. So these are all required for PCR. Now let's go to start the steps of PCR, that how the PCR can be done. So the first step is denaturation. So in the denaturation, we have a double-stranded DNA. So in this step, this double-stranded DNA are separate from each other like this. This become one template strand, and this is become another template strand. So we do temperature to this double-stranded DNA. So the temperature are be 95 degrees centigrade to denature this double-stranded molecule and two template strand. Now when the two template are formed, then the next step are started. In first step, the double-stranded DNA are converted into two template strand. Now in annealing, so annealing are basically those process are those step of PCR in which primer attached to these template strand. So one pair of primer are required for the annealing steps. So I want to draw the diagram. So this is the one template strand, and this is the another template strand. So one primer attached to this side and another primer are attached to this side. Now when the primer are attached, so here the temperature are 54 required. When we do a denaturation process, we make two template strand. So we reduce the temperature from 95 to 54 degrees centigrade. In the annealing steps, the primer are attached to our template strand. Now when primer are attached, then the polymerization steps are started. So what here done, the DNTPs, which are present in our master max. So they attach to this primer one DNTPs are coming and attached to this our template strand and make a double stranded DNA. So like that, this is our one strand, and this is the another strand. And the polymerization step, we increase the temperature from 54 to 72 degrees centigrade, because the DNA polymerase do its activity on 72 degrees centigrade. Now, when we increase the temperature to 72 degrees centigrade, the uh, DNTPs are the tag polymerase attach DNTPs which are present in our master mix. They attach one by one uh, uh, nucleotides and make a double stranded DNA like that. Polymerase do its work and add its nucleotide and make a double stranded DNA molecule. Also here. So two DNA strand or two DNA molecule are produced from a single DNA molecule. So this is the one cycle of polymerase chain reaction. And another cycle, these two DNA molecule are separate, denaturation occur, and then the annealing, the primer are attached, and then the deep polymerization are occurred. So the graphical representation of this DNA polymerase chain reaction is here. First, we increase the temperature. First, we increase the temperature to 95 or 94 degrees centigrade. Then we reduce the temperature to 54 degrees centigrade. Then we increase our temperature and to 72 degrees centigrade. So this is the graphical presentation of PCR polymerase chain reaction process. And first step in denaturation, we increase the temperature to 95 degrees centigrade. Then we in annealing, when primer are attached, so we reduce temperature to 54, and when primer are attached, so we increase the temperature to activate our D take polymerase enzyme to 72 degrees centigrade. Now let's go to the application of PCR, that and what in which area we use the polymerase chain reaction. So the first one application of PCR is DNA fingerprinting. So in DNA fingerprinting we use PCR process. And diseases diagnosis, we can also diagnose different type of disease like hepatitis B virus, COVID, uh, coronavirus, and also other disease. The we also use PCR for species identification, that we identify different type of species using PCR. And we can also identify the migration tracking of animals, and we also can find out quantification of nucleic acid. So hopefully you understand this lecture. uh, Thank you, see you next time.